Dear students, today we will study polyploidy in plant breeding. This is also an important topic of your course. History. The first example of an artificial polyploid was by Winkler in 1916, who in fact introduced the term polyploidy. Winkler was working on vegetative graft and chimeras of Solanum nigrum and found that callus regeneration from cut surface of the stem explant were tetraploid. One of the early examples of natural polyploid was one of the original mutants of Ornithera lamarcania. What is ploidy? Ploidy refers to the number of basic chromosome sets. For example, a diploid has 2 sets, where a hexaploid has 6. The basic chromosome number X is also called the monoploid number. So, monoploid is the number of different chromosomes that make up a single complete set. Ploidy can be divided into 2 groups based on the type of change in chromosome number, that is euploidy and aneuploidy. Euploidy, the condition of an organism having complete set of chromosome or multiples of the basic set is euploidy, and the cell is said to be euploid. Aneuploidy, this is a condition where the chromosome number is not an exact multiple of the normal diploid number, with either fewer or more than the normal number of chromosomes in the cell. The condition of an organism having a set of chromosomes or multiples of basic set is euploidy, and the cell is said to be euploid. Euploidy can be divided into monoploidy or polyploidy. Monoploidy means single set of chromosome and polyploidy means multiple set of chromosome. For example, 3x, 4x, 5x, 6x, etc. Euploidy is lethal in most animal species but often tolerated in plants, where it has played a role in speciation and diversification. Monoploidy and polyploidy involve complete set of chromosome, and so both are cases of euploidy. Causes of occurrence of monoploidy and polyploidy. Monoploidy and polyploidy can result when either round of meiotic division lacks cytokinesis or when meiotic non-disjunction occur for all chromosomes. It can occur when complete non-disjunction at meiosis 1 will produce half gamete with normal chromosome and half with no chromosome, or a gamete with 2 sets of chromosome fused with a normal gamete produce a triploid zygote. Fusion of 2 gametes that each have 2 sets of chromosome produces a tetraploid zygote. Polyploidy of somatic cell can result from mitotic non-disjunction of complete chromosome sets. Monoploidy, an individual that contains one half of normal number of chromosome is a monoploid, and it exhibits monoploidy. Monoploids are rare in nature because recessive lethal mutations become unmasked and they die before they are detected. These alleles normally are not a problem in diploids because their effects are masked by dominant allele in the genome. Monoploidy has been applied in plant biotechnology to rapidly develop plants from anther that have a fixed genotype. Monoploids are often useful in plant experiments. For example, haploid cells are isolated from plants. Colchicine, which inhibit mitotic spindle formation, allow chromosome number to double producing completely homozygous diploid breeding line. Mutant genes are easily identified in monoploid organisms. In this slide, you can see production of monoploid plants from diploid. Here, the plant diploid plant is taken and its anther is collected. Mitotic products are plated. Okay. Haploid embryos grow. Now, haploid, the cell is grown by tissue culture as haploid embryo. Now, haploid plantlets, and it is planted again. The plant is now monoploid. This is an easy technique applied by genetic engineers. Polyploidy. Polyploidy occur in cells or organisms when there are more than two homologous set of chromosomes. It is very common in plants. It is associated with origin of new species. Polyploid is common in nature and provide a major mechanism for adaptation and speciation. It may be positive correlate to size of the individual. You can see polyploidy usually increase the size of an organism or fruits. There are two classes of polyploids based on the number of chromosome sets. Even number polyploids and odd number polyploids. Even number polyploids are more likely to be at least partially fertile because the potential exists for equal segregation of homologs during meiosis. While odd number polyploids will always have unpaired chromosome, balanced gametes are rare and these organisms are usually sterile or have increased zygote depth. Let us see in this diagram that diploid, pentaploid, all with odd number of chromosome set are often sterile due to pairing problem. Here you can see a diploid. Okay, the pairing possibility is that here it may be 2 chromosome and here it may be 1. It is bivalent plus univalent. Even number of chromosomes in polyploid may lead to normal segregation and partitioning of the chromosome set. For example, here you can see that it is 4 chromosome or tetraploid and we can get 2 bivalent, or pairing possibility is also 1 quadrivalent we can get, and we can also get 1 univalent or trivalent. Examples of polyploid. Let us see. Common example is maize, and it has polyploidy 2x20 and it is diploid. It is all outcrossing. Wheat, it is hexaploid and it is propagated by outcrossing. Rice, it is diploid and it is propagated by self-help. Potato, it has 4x is equal to 48. It is tetraploid and it grows by outcrossing or vegetative reproduction. Maize, soya bean, it is 2x is equal to 40 and it is diploid. It is propagated by selfing. Barley, it is 4x is equal to 14 and it is diploid and it is propagated by selfing. Potato, 2x is equal to 24, diploid and it is propagated by selfing. Banana, it is 3x is equal to 33. It is diploid and it is propagated by vegetatively. Watermelon, it is 2x is equal to 22. It is diploid and it is propagated by outcrossing. Sugarcane, it is 8x is equal to 80. It is octaploid and it is grown by outcrossing and vegetative propagation. Sugar beet, it is 2x, that is 18. It is diploid and it is grown by outcrossing. Cassava, it is 2x is equal to 36. It is diploid and it is propagated by outcrossing or vegetatively. Characteristic of polyploids. Plants are generally bigger in size or stature. The plants are especially sterile if they have odd number of chromosome sets. Polyploidy tolerated in plants because of self-fertilization plays an important role in plant speciation and diversification. Polyploidy is of two types. Autopolyploidy and allopolyploidy. Autopolyploidy. Autopolyploids result when all set of chromosomes are from the same species, usually due to meiotic error. Containing of multiple copies of the basic set of chromosome of the same genome occurs in nature through union of unreduced gamete. Natural autoploids include tetraploid crops such as alfalfa, peanut, potato and coffee, and triploid bananas. In this slide, you can see autopolyploidy. Here you can see the parent are diploid, two set of chromosome are here, and here it is polyploid. It is autopolyploid because all the set of chromosomes are of same type. Autopolyploidy can arise spontaneously through non-disjunction of a whole chromosome set. A triploid commonly made by crossing diploid to tetraploid, or tetraploid can be induced by treatment of colchicine. For example, seedless fruit like banana, that is 3n is equal to 33, grapes and watermelon, they are seedless and they are example of autopolyploid. Grasses, garden flower, crop plants and forestry are also examples. Allopolyploidy. Hybrids of two or more closely related species. Partially homologous chromosome, that is homeologous. Allopolyploidy is caused by presence of several genomes of different species in a cell, a tissue or a whole organism. That is a combination of genome from different species. They result from hybridization of two or more genomes followed by chromosome doubling or by the fusion of unreduced gametes between two species. This mechanism is called non-disjunction. These meiotic aberrations result in plants with reduced vigor. Economically important natural polyploids crop include strawberry, wheat, oats, upland cotton, oilseed rape, blueberry and mustard. Let us see all allopolyploidy result from viable mating of two different species. Here it is species 1 and it is species 2. In species 1, if there is meiotic division and normal gametes are produced, and in species 2, polyploid gametes are produced. If they both are crossed, then in first meeting we get polyploid gamete, which is of two different types. It is from species 2 and it is from species 1. Okay. Again, when we made this polyploid gamete with normal gamete, what can happen? In second mating, we can get polyploid. Here, each set of chromosome is complete and it is a polyploid and it is allopolyploid of a chromosome set with two different type of genomes. For example, fusion of haploid gametes from species 1 and species 2 produces an N1 plus N2 hybrid plant. No chromosomal pairing occur at meiosis, viable gametes are not produced and the plant are sterile. Rarely, division error or cold shock doubles the chromosome set, that is 2n1 plus 2n2. The diploid set function normally in meiosis and fertile allotetraploid plant result. An example is cross between cabbage, that is Brassica oleracea, and radish, that is Raphanus sativus, which both have a chromosome number of 18. If pairing partner can be provided for homologs in allopolyploid, sterility can be revived. For example, Raphanus. Here you can see Raphanus, it is the plant. It is called amphidiploids because of non-identical chromosome set double in alloploids. Here you can say that in reddish or Raphanus sativus, n is equal to 9, and in cabbage also n is equal to 9. When we cross this 2n plus 2n, the Raphanus is tetraploid. It is it can be called amphidiploid, and its chromosome number is 4n is equal to 36. Artificial allopolyploids. Artificial or synthetic allopolyploids have been synthesized in some crop with two main objectives. Number one, either to study the origin of naturally available alloploids or to explore the possibilities of creating new species. Polyploidy is the rule in agriculture, where polyploids include all commercial grains. For example, bread wheat, that is Triticum acetivum. It is 2n is equal to 42. It is an allohexaploid of 3 plant species. Most crops and common flowers are polyploids. Hybrid vigor resulting from interspecific process in allopolyploids is one of the most exploited advantages of polyploidy in plant breeding. Let us see how natural polyploids evolved in bread wheat. A variety Triticum monococcum, which is coded by 2A, having 14 chromosomes, was crossed with wild variety of Triticum of a known species, which was also 14 chromosome. It was in nature, and the sterile hybrid AB occur. It contained 14 chromosomes. But when spontaneous chromosome doubling occurs, then a variety Triticum turadium occurs, which contain 28 chromosomes. It is represented by AA and BB. Okay. When this Triticum turadium is crossed with another variety Triticum tushai, which contain 14 chromosomes, then a sterile hybrid A, B and D was produced and containing 21 chromosomes. Again, when spontaneous chromosome doubling occur, then another variety AABBDD occur. That is hexaploid, and it is Triticum acetivum. It contains 42 chromosomes and it is our bread wheat. Here in this diagram, it is explained how allotetraploid occur. This is species 1. It contains 2n, that is AA. A haploid AN is pollen, and AA is also occur, that is 2n, it is pollen. A species 2 that has 2n BB and an ovule, it is N ovule, and BB is 2n ovule. Okay, one is haploid and one is diploid. Okay, when it is crossed, when these species 1 and species 2 are crossed, then we get allotetraploid. Here 2n is equal to AA and BB. Okay, first step. And now what happened when triploid bridge occur? When AA and BB is crossed, it get normal 2n species will be AABB. But when triploid bridge occur, it means when 2n pollen of one and n ovule of another species is crossed, then what happen? 2n is equal to AAB. Okay, this is a sterile plant, and this is when crossed with only B, a haploid plant, then we can also get 2n, that is AABB. It will be fertile. Again, hybrid bridge. If A haploid species is crossed with B, then we get 2n is equal to AB. And now when AB is further crossed, multiplied, we get 2n is equal to AB. This will be fertile. And somatic doubling. If A plus B gets 2n AB, then also we get 2n, that is AABB. These will be fertile and these triploid and these will be unfertile. Let us see difference between autopolyploidy and allopolyploidy. Autopolyploidy refers to a type of polyploid where the chromosome complement consists of more than two copies of homologous chromosome. It is arisen by fusion of gametes of the same species. An autopolyploid cells contain homologous chromosome set. It is mainly caused by non-disjunction of chromosome. It does not undergo meiosis since it has arisen from one parent. It is seen in crops such as wheat, oat, sugarcane, potato, peanut, banana, and coffee. In allopolyploidy, it refers to the type of polyploid where the chromosome complement consists of more than two copies of chromosome derived from different species. It is arisen by fusion of gametes of different species. All the chromosome set of the allopolyploid are not homologous. It is caused by mating of different species. It can undergo meiosis and it is seen in crops such as wheat. Aneuploidy. This is a condition where the chromosome number is not an exact multiple of the normal diploid number, with either fewer or more than the normal number of chromosome in the cell. Aneuploids are polyploid that contain either an addition or subtraction of one or more specific chromosome to the total number of chromosomes that usually make up the polyploidy of the species. Causes. A condition of aneuploidy may occur due to loss of one or more chromosome during mitosis or meiosis, or due to non-disjunction, that is irregular distribution of sister chromatin. Let us see how aneuploids originate. These are two chromosomes. When non-disjunction occur in first cell division, then what happens? One set of chromosome has gone to a cell and another is empty. Then when it is further having meiosis division, then what happened? N plus 1 and N plus 1 in this, and in this N minus 1 and N minus 1. Okay. And another case, if there are two cells, okay, when there is normal division, no disjunction, okay. And in this case, when N plus 1 occur in 1 and N minus 1 occur in this non-disjunction occur, and in this if only 1 and 1 get, okay, then we get aneuploids, which is less than normal chromosome. In diploid organism, aneuploid variations take four main forms that can be classified under two main groups. Number one is hypoploidy and number two is hyperploidy. Loss of one or more chromosome from the basic set is hypoploidy, and presence of one or more chromosome in the basic set is hyperploidy. Aneuploidy is divided into hypoploidy and hyperploidy. And it can be, hypoploidy can be monosomic, that is 2n minus 1, or nullisomy, that is 2n minus 2. And hyperploidy can be trisomy 2n plus 1, tetrasomy 2n plus 2, pentasomy 2n plus 3, and it can be so on. Monosomy. It is a form of aneuploidy with the presence of only one chromosome from a pair. A monosomic cell hence involves a loss of single chromosome, that is, the cell is 2n minus 1. Partial monosomy occur when only a portion of the chromosome has one copy while the rest has two copies. This condition mainly occurs due to the non-disjunction in meiosis in parent producing a gamete with no copies of a particular chromosome and one copy of all other chromosomes in the cell. The number of possible monosomics in an organism is equal to the number of chromosomes. Monosomy is usually not tolerated in diploids but can be easily introduced in polyploids. However, Several experiments have been carried out and certain degree of success has been observed. Usually, autosomal are lethal due to dosage imbalance. Nullisomy. It involves loss of one homologous chromosome pair, that is the cell is 2n-2. It is lethal in diploid but can be tolerated in bred wheats because it is a hexaploid. It behaves meiotically like a diploid. It has 4 homologous chromosomes. It is compensated for a missing pair of homologous. Sear isolated 21 nullisomics in wheat. It occurs mainly due to non-disjunction occurring for the same chromosome in meiosis in both parents, producing gametes, each with no copies of that chromosome and one copy of all other chromosomes in the set. Trisomy. A chromosomal abnormality that is characterized by presence of an extra chromosome in the cell of a plant body. A trisomic cell involves an extra chromosome, that is, the cell has 3 copies of one chromosome type and 2 copies of every other chromosome type. A trisomic cell is 2n+1. A partial trisomy occurs when part of an extra chromosome as opposed to a whole extra chromosome occur. Trisomics are viable. They may be fertile and can produce viable meiotic products. Tetrasomy. The condition of a tetrasomic cell involves an extra chromosome pair resulting in the presence of 4 copies of one chromosome and 2 copies of every other chromosome type. A tetrasomic cell is hence 2n+2. This is caused by non-disjunction for the same chromosome in meiosis in both parents, producing gametes each with 2 copies of that chromosome and 1 copy of all other chromosomes in the set. Let us see some terminology applied in polyploidy breeding. Heteropolyploidy. Individual carrying chromosome number other than the diploid. Aneuploidy. Change in one or few chromosome of the genome. Nullisomic. One chromosome pair missing, that is 2n-1. Monosomic, that is one chromosome missing, that is 2n-1. Double monosomic, if one chromosome from each of 2 different chromosome pair missing, that is 2n-1-1. Trisomic, if one extra chromosome present, that is 2n+1. Tetrasomic, if one extra pair of chromosome present, that is 2n+2. Euploid involves one or more complete genome. Monoploid, one copy of a single genome. Haploid, gametic chromosome complement. Polyploid, more than 2 copies of genome. Autopolyploid, genome identical with each other. Autotriploid, 3 copies of one genome, that is 3x. Autotetraploid, 4 copies of one genome, that is 4x. Allopolyploid, 2 or more distinct genome. Allohexaploid, 3 distinct genome. Breeding of autopolyploids. Cells or individual having autopolyploid chromosome number may originate in one of the following ways. Maybe it may be spontaneous, it may be due to treatment of physical agent, it may be due to regeneration in vitro, it may be due to colchicine treatment or other chemical agents treatment. Spontaneous. Chromosome doubling occur occasionally in somatic tissue and unreduced gametes are also produced in low frequencies. Production of adventitious buds. Decapitation in some plants lead to callus development at the cut end of the stem. Such a callus has some polyploid cell and some of the shoot buds regenerated from the callus may be polyploid. This is of common occurrence in Solanaceae, where 6 to 36% of the adventitious buds are reported to be tetraploid. By physical agent, heat or cold treatment, centrifugation, X-rays or gamma rays, irradiation may produce polyploid in low frequencies. Tetraploid branches were produced in Dhatura in response of cold treatment. Polyploidy is a common feature of the cell culture in vitro. Plants of various ploidy have been regenerated from callus culture of Nicotina, Datura, rice and several other species. Colchicine treatment. Colchicine treatment is the most effective and the most widely used treatment for chromosome doubling. At any given time, only a small portion of the callus would be in division. Therefore, repeated treatment should be given at brief intervals to double the chromosome number in a large number of cells of the shoot apex. The polyploid and diploid cells may often outcompete the polyploid ones. Applications of polyploid. It is bridge crossing, creation of new crop species, interspecific gene transfer, tracing of origin of species, mutation breeding, seedless fruit, ornamental and forage breeding, disease resistant through aneuploidy and industrial application of polyploids. Bridge cross. Amphidiploid can be used as a bridge cross where direct cross between two species is not possible and died due to sterility in F1. Creation of new species. Alloploidy sometimes help in creation of new crop species such as Triticale, Rapinobrassica, etc. Interspecific gene transfer. In case of unavailability, the desirable character within the species, it is transferred from the related species. Tracing the origin of crop species. Alloploidy study is used to identify the origin of natural alloploidy plants. Mutation breeding. Polyploids are able to tolerate deleterious allele modification post-mutations. They have increased mutation frequency because of their large genome resulting from duplicated condition of their gene. Next is seedless fruit. No isolation, sterile hybrid formed. It occurs when a haploid gamete fuses with a diploid gamete, resulting in a triploid plant which is sterile. For example, polyploid banana watermelon. Ornamental and forage breeding. Polyploids in plant is an increase in cell size. This phenomenon is termed as Giga's effect. Ornamental crops such as snapdragon and marigold have been bred through chromosome doubling to improve the quality and size of their blossom. The slower growth rate of polyploid allow them to flower later and for a long period of time than the diploid progenies. This quality may be interest especially in ornamental breeding. Disease resistance through aneuploidy. Aneuploidy applied in plant breeding to produce disease-resistant plant through the addition of one extra chromosome in the progeny genome. For example, the transfer of leaf rust resistance to Triticum acetivum from Aegilops umbiliculata through backcrossing. In addition, other breeding strategies utilize aneuploidy have been explored, including chromosome deletion, chromosome substitution, and supernumerary chromosome. Next is industrial application of polyploidy. Commercial synthesis of sex hormones and corticosteroids have been improved significantly by artificial induction of tetraploids from diploid Dioscorea gingivirensis native to China. Other plants whose production of terpenes has increased following artificial chromosome doubling. It enhances production of secondary metabolites such as alkaloid-centered beans in polyploidy. Many concurrently offer resistance to pests and pathogens. Significance of polyploidy in plant breeding. It permits greater expression of existing genetic diversity. It helps to change the character of a plant by altering number of genome, consequently changing dosage of allele related to a particular trait. Polyploids with uneven number of genome like triploid may result in infertility. This loss of seed production can be used to produce seedless watermelon and bananas. About one-third to half of the angiosperms are polyploid. About 70% of wild species of grasses and 23% of legume family are polyploids. Most of the natural polyploids are allopolyploids. However, all species doesn't exhibit vigor with increased in polyploidy. For example, optimum ploidy level in per corn is diploid as compared to tetraploid, and optimum ploidy level of banana is triploid, that is seedless banana. Advantages of polyploid: enlargement and increased vigor, creation of a sterile triploid, restoring fertility in hybrids, overcoming barrier in hybridization, and enhancing pest resistance and stress tolerance are some of the advantages of polyploid. With the knowledge of the origin, variation, and characteristic of different types of polyploid, there are many opportunities for developing and utilizing polyploids in plant improvement programs. Significant opportunities including developing sterile cultivar, overcoming barrier to hybridization, restoring fertility in wide hybrids, enhancing flower size, increasing heterosis and vigor, and improving pest resistance and tolerance to environmental stress. These are some suggested readings. Please read these books. That is all for today. Thank you.